The era of the modern synthesis in the early 1900s brought together evolutionary biology, genetics, and math. The mathematics help us determine when allele frequencies are changing, which is when evolution is occurring. The Hardy-Weinberg equation is in equilibrium when mating is random, without selection, drift, flow, or mutation. So when Hardy-Weinberg equation is not in equilibrium, we know some selection, drift, flow, or mutation must be occurring. In 1908, a British mathematician, G.H. Hardy, and a German doctor, Wilhelm Weinberg, each published on this work. At the time, it was believed that changes in allele frequency occur simply as a result of sexual reproduction. Meiosis followed by the random fusion of gametes to form offspring. Some biologists claim that dominant alleles inevitably increase in frequency when gametes combine at random. Others predicted that two alleles of the same gene inevitably reach a frequency of 0.5. To test these hypotheses, Hardy and Weinberg analyzed the frequencies of alleles when many individuals in a population mate and produce offspring. Instead of only thinking about the mating between two parents with a specific pair of genotypes, they looked at what happened when an entire population, when all of the individuals and all genotypes bred. Like Darwin, Hardy and Weinberg were engaged in population thinking. A population is a group of individuals from the same species and time that can interbreed and may vary in traits. To analyze the outcomes of matings among all individuals in a population, Hardy and Weinberg imagined alleles from all gametes in a generation going into a gene pool and combining randomly, like what happens when sea stars release their gametes into the water before combining to form zygotes. They applied math to determine which genotypes would be present in the next generation and in what frequencies. Calculating what happens when gametes from the gene pool combine randomly to form offspring. Their calculations predict the frequency of genotypes of the offspring that would be produced.
Hardy and Weinberg began by analyzing a simple situation with just two alleles of a particular gene. I know this doesn't look that simple, so let's get more familiar with the calculations for alleles A1 and A2, deriving it empirically, to build our intuitive familiarity with this useful mathematical principle. The Punnett square shows the outcome of random mating, the random combinations of all gametes in a population. The letter P is used to symbolize the frequency of A1 alleles in the gene pool, and Q is used to symbolize the frequency of A2 alleles in the same gene pool. With only two alleles, their frequencies must add up to 1. Therefore, P plus Q equals 1, since they are 100% of the cases. The frequency of the A1A1 genotype is P squared. The frequency of the A2A2 is Q squared. And the frequency of A1A2 is 2PQ. Although P and Q can have any value between 0 and 1, let's suppose that the initial frequency of A sub 1 is 0.7, and that of A2 is 0.3. In this gene pool, 70% of the gametes carry A1 and 30% carry A2. In general, there is a P chance that it carries A1 and a Q chance that it carries A2. So here, a gamete forming an offspring has a 70% chance that it carries A1 and a 30% chance that it carries A2. With two alleles present, 3 genotypes are possible in the offspring generation: A1A1, A1A2, and A2A2. What will be the frequencies of these 3 genotypes? According to the logic of Hardy's and Weinberg's result, the frequency of the A1A1 genotype is P squared. The frequency of the A1A2 genotype is 2PQ. The frequency of the A2A2 genotype is Q squared. Since genotype frequencies in the offspring generation must add up to 1, this means that P squared plus 2PQ plus Q squared equals 1. Given our example frequencies of 0.7 and 0.3, This simplifies to 0.49 plus 0.42 plus 0.09 equaling 1. This is the Hardy-Weinberg equation, and you can become more comfortable with it by visualizing it in the real world and then practicing. Imagine all the parental clams in a generation of a population are sitting randomly in a gene pool of seawater. All the gametes from A1A1 clams carry A1, so 49% of the gametes in the gene pool will carry A1. Half of the gametes from A1A2 will also carry A1, so an additional 21% of gametes in the gene pool carry A1, for a total of 49% plus 21% equaling 70% A1. The same logic applies to determining the frequency of A2. Notice the frequency of allele A1 in the offspring generation is still 0.7, and the frequency of allele A2 is still 0.3. Thus, the frequency of allele A1 is still P, and the frequency of allele A2 is still Q. No change in allele frequency occurred. Even if A1 is dominant to A2, it does not increase in frequency. And there is no trend toward both alleles reaching a frequency of 0.5. This result is called the Hardy-Weinberg principle. The Hardy-Weinberg principle makes two fundamental claims. If the frequencies of allele A1 and A2 in a population are given by P and Q, then the frequencies of genotypes A1A1, A1A2, and A2A2 will be given by P squared, 2PQ, and Q squared, respectively, for an arbitrary number of generations carrying onward in the Hardy-Weinberg equilibrium. Alleles transmitted via meiosis and random combinations of gametes have frequencies that don't change over time. Then for evolution to occur, some other factor that disturbs this equilibrium must be happening. The Hardy-Weinberg model is based on important assumptions about how populations and alleles behave. These assumptions help to define the four processes of evolution that can be acting on the population. Selection, whether artificial or natural, gene flow, genetic drift, or mutation. Here are the 5 assumptions that must be met for the Hardy-Weinberg equation to be in equilibrium. The model assumes that mating is random with respect to the gene in question. Visualizing this, imagine the gametes in the gene pool float around in the seawater randomly, not choosing mates. The model assumes that there is no natural selection, so all members of the parental generation survive and contribute equal numbers of gametes to the gene pool, no matter what their genotype. The model assumes no genetic drift, so alleles in exact frequencies P and Q are combined and drifting by chance. Drift occurs by chance when populations are small, since tossing a coin only 10 times might produce unexpected results. But tossing 1,000,000 times will come closer to expectations, so the model is assuming infinitely large population. In our example, allele A1A1 did not get lucky and get drawn more than the expected 70% of the time. The model assumes no gene flow with no new alleles being added by immigration or lost through emigration. That is, that all of the alleles in the offspring population came from the original population's gene pool. The model assumes no mutation, such that no new alleles are introduced into the gene pool by mutation. The Hardy-Weinberg principle tells us if selection, genetic drift, gene flow, and mutation are not affecting a gene, and if mating is random with respect to that gene, under these conditions, the genotypes A1A1, A1A2, and A2A2 should be in the Hardy-Weinberg proportions, and no evolution will occur. If Hardy-Weinberg is not in equilibrium, then evolution is occurring. Biologists often want to test whether natural selection is acting on a particular gene, or non-random mating is occurring, or one of the other evolutionary processes is at work. In addressing questions like these, the Hardy-Weinberg principle functions as a null hypothesis. Recall, a null hypothesis predicts there are no differences among the treatment groups in an experiment. The Hardy-Weinberg principle predicts that genotype frequencies will occur when mating is random with respect to that gene, and when natural selection, genetic drift, gene flow, and mutation affecting the gene. If biologists observe genotype frequencies that do not conform to the Hardy-Weinberg prediction, it means that something interesting is going on. Either non-random mating is occurring, or allele frequencies are changing for some reason. Further research is then needed to determine which of the 5 Hardy-Weinberg conditions is being violated.